Kingdom Monera, the next group are the Eubacteria. Eubacteria are the organisms which are considered to be true bacteria, and these are, as we know, these are unicellular and prokaryotic in its nature. Some of these bacteria are photosynthetic, but majority of them are heterotrophic, but few of them are autotrophic, that is photosynthetic in their nature. Actually, the bacteria have been first discovered by Anton von Leeuwenhoek, and he named this bacteria as animalcules. So, when he discovered this microscopic organisms, he come to know that some type of dazzling movement was there. When he was observing his scrapings under the microscope, he found that some type of movement was observed. So, that random or dazzling movement was there. Because of that one, he called them as animalcules. But later, Ehrenberg had given the term bacteria to those animalcules. So, that is the history how this bacteria have been identified, and the name have been come to those microscopic living organisms, that is bacteria. And where does this bacteria occur? Bacteria ubiquitous. Ubiquitous means these are present everywhere, elsewhere they are present. So, there is no restricted habitat for them. So, they are cosmopolitan, that is they can present in each and every place on the earth. That is they can present inside the soil, in the water, and in the atmospheric conditions. They can survive in hot springs, sulfur hot springs, and also they can survive in the very cold conditions like in the snow also. So, that is no such restricted habitat for these organisms. That is why these are ubiquitous, and they can survive in alkaline, they can survive in acidic, they can survive in salty nature. So, there is no restricted side. Acidic nature, they can survive in acidic conditions, alkaline conditions, hot sulfur springs, saline conditions. They can survive in very low temperatures, cold conditions. So, they can survive in such type of harsh and so critical conditions also. This type of occurrence is there related to the bacteria. Bacteria, if we observe the other characteristic features related to bacteria, bacteria are showing the difference in their structural relationship and the shape. Let us start with the first, the shape. So, bacteria exhibit lot of morphological variation. So, based on the shape, the bacteria have been divided into coccus bacteria. Singular is coccus and plural is cocci. The spherical shaped bacteria are called as coccus bacteria, and these coccus bacteria will exist in different forms, like one is monococcus. Mono means single, coccus means spherical. So, only single spherical bacteria will be present. The second one is the diplococcus. Diplo means two. Two spherical shaped structures are present in the bacteria. Third one is tetracoccus. That is four spherical shaped structures are present. Fourth one is streptococcus. Strepto means many spherical shaped structures are present, but they are in the form of a chain. A chain of spherical structures is present in the streptococcus, like this. And fifth one is the staphylococcus. Here also many spherical shaped structures are there, but their organization is not in a definite pattern. Like this, the arrangement will be in a irregular manner. So this type of organization is called as a Staphylococcus. Then sixth one is Sarcina. Sarcina is an arrangement of 8 coccus structures which are in the form of a cube. So 4 can be seen on one side and the remaining 4 will be present on the other side or back side like this. So cube will be formed. So cube of 8 spherical structures. So like in the spherical itself, we can see based on the arrangement pattern. So 6 types of coccus bacteria are present. The second type in the shape or the Bacillus or bacilli. Bacilli means the rod shaped bacteria. And in this bacillus also, based on the number of the rod shaped structures, they have been divided into one monobacillus. That is a single rod shaped structure is present. This is called as a monobacillus. The second one is a diplobacillus, which contains 2 rod shaped structures. And third one is a streptobacillus, in which a chain of rod shaped structures are present. So like this, based on the arrangement or the number, the bacillus are of 3 types: monobacillus, diplobacillus and the streptobacillus. In the shape, the third type are Spirulum. So Spirulum are the spiral shaped bacteria. So they show some sort of coiling, little bit spiral organization. So they have the flagella on both the sides. So this type of structures are called as a Spirulum. And some are called as spirochete are there. In this only spirochetes are there, which are flexible. The flexible Spirulum are called as a spirochete. Example is Cristispira. And the fourth one is Vibrio. Vibrio are the comma shaped bacteria. So the structure appears to be like this. So this type of shapes are being exhibited by different groups of bacteria. But at the same time, there are some bacteria which will exhibit some special feature. That is, they have the ability to change their shape and based on the nutrition available and the environment in which it is present. Pleomorphic bacteria. There are some bacteria which will change their shape based on the environment in which it is present and also the nutrition that is available to them. So based on these two characters, if a bacteria is changing its shape, then we call it as a pleomorphic bacteria. Example for this one is Acetobacter. Acetobacter. So here this Acetobacter will exist in different shapes or different forms. That may be, it may be in the form of small chains or large chains or in different structures also. That's what it is going on, changing its shape or the form based on the environment and the nutrition which is available to it. Such type of special characteristic features are present in the pleomorphic bacteria. Example is Acetobacter. Next one is a bacterial cell structure. Structure of bacterial cell. Here in the structure of bacterial cell, basically three parts it have been divided. One is structures external to cell wall. Second one is cell wall, and third one is structures internal to cell wall. So that is a structure of bacterial cell is divided into, or can be studied in an easy manner by dividing it into three parts. So let us start with the first one, the structures external to the cell wall. Generally we know that cell wall is the outermost part in the plants, but here in bacteria cell wall is not the outermost limiting layer, but outside that cell wall some other secretions are there which form into a layer like structure. So we call such type of structures, and some extra appendages will also be formed. Those are also been the part of the structures external to the cell wall. Those include glycocalyx, flagella, pili, sex pili like structures. Let us start with the first one, the glycocalyx. Here if we consider the bacteria like this, this is a cell wall of the bacteria, and outside this one we can see some additional layer. This additional layer is called as a glycocalyx. This glycocalyx is made up of polysaccharides. And this have the special feature, that is it is slimy and gelatinous. In the composition, glycocalyx contains polysaccharides and amino acids, and it is slimy and gelatinous in its nature. It have the dual nature that it can exist in the form of a loose sheath. When it is in the form of a loose sheath, we call it as a slime layer. Sometimes it is firmly attached to the cell wall. When it is firmly attached, we call it as a capsule. Whether it may be in the form of slime layer or the capsule, its composition will be made up of polysaccharides and the amino acids. So here this nature of loose sheath or firm nature is genetically controlled. And what is the function of the glycocalyx? Generally, it will give protection to the bacterial cell and also protect it from desiccation, and also protect it from external harmful reagents or harmful atmospheric conditions. So that is the way how this glycocalyx will always be protecting the bacterial cell. Apart from this one, next part is a flagella. Here from inner side of this one, or inner side to the cell wall, some special structures which are arising from the bacteria cell. This flexible structure is called as a flagella, and the flagella is made up of a protein called as flagellin. This flagella, when we take the transverse section, it should have 9+2 arrangement in the eukaryotic organisms, but as bacteria is an prokaryotic organism, here 9+2 arrangement is not present in the transverse section. Actually, what do you mean by this 9+2 means? In the transverse section, we can see that 2 central fibrils will be there and 9 peripheral fibrils are present. But here the 2 central fibrils are lacking, only the 9 peripheral fibrils are present. So that's the reason why 9+2 arrangement is not exhibited by the flagella of bacteria, as it is not a eukaryotic one, but it is a prokaryotic organism. And this the long and flexible structure it is there, which is made up of axoneme, and this axoneme will arise from a structure called as a basal body. Here the basal body is present below the cell wall or the plasma membrane. From there the axoneme will arise, and connecting the basal body to the axoneme, here a hook-like structure will be present. So these are the 3 structures or the parts which can be seen in the flagella, and this flagella will help in the locomotion, or it is a mobile organ or motility. It is helping in the motility or the locomotory structure, and here it will help in locomotion by rotating, that is pushing the structures in the rotation manner such that it will help in the locomotion of the bacteria. So this is about the structure called as flagella, which are long and flexible. Here all type of bacteria except the coccus. Coccus one lack the flagella, and some may have one flagella, some may have more than one also. Based on the number and distribution of flagella, the bacteria are classified into 5 types. Those 5 types are Atrichus. Atrichus means the bacteria which completely lack flagella. So the best example for the group of bacteria which does not have the flagella or the coccus bacteria. So here the example is Lactobacillus is other example where flagella are completely absent. Of course, Lactobacillus will belongs to Bacillus type, and the coccus or all spherical bacteria lack flagella. And here if you see the diagram of this Atrichus ones, it appears to be like this. So bacterial structure without any flagella on its surface. Then second one is Monotrichus. Mono means single, one flagella. Is one, and it is present on one surface or the one pole. Only one side the flagella will be present. So this can be seen in example like Xanthomonas. And third one is Lophotrichus. Lophotrichus means a tuft of flagella or a group of flagella present on one side or one pole. So here Lophotrichus can be seen in the example. The diagrammatic representation will be like this. Example is Spirulum. The fourth one is Amphitrichus. Here in Amphitrichus, the flagella are present on both the sides or both the poles. This type of bacteria with flagella are called as Amphitrichus. So here in this diagrammatic representation, we can see the flagella on both the surfaces. So Amphitrichus can be seen in Nitrosomonas. The fifth type is the Peritrichus. Here many flagella are present, which are present throughout the surface of the bacteria. Like if you see the diagrammatic representation, this is a bacterial structure with a flagella on all the sides. So this can be seen in examples like E. coli, Escherichia coli and Salmonella. So likewise, based on the distribution and the number of flagella, 5 types of bacteria are present. In the structures external to the cell wall, we can see some other structure called as pili or fimbriae. So if you observe the bacterial cell, outside this bacterial cell, we can see some other extruded structure apart from the flagella. Those extruded structures are small but stiff. These small and stiff structures are called as pili or fimbriae. So how can we differentiate the flagella from pili? Means here these are comparatively short, stiff, and their number is more when compared to the flagella. So that is the way how we can differentiate the pili from the flagella, and these are made up of a protein called as pilin or fimbrillin. Generally these pili have solid core, that is central part of this one will be without any hollow structure, that is entire, it is filled with the materials, and the function of this pili or fimbriae is useful for attachment of bacteria to the substratum. Either to a terrestrial substratum or the aquatic substratum. So that means the pili are differentiated from the flagella in terms of their structure, nature and function. And in the pili itself, some special type of pili are called as sex pili. So how does the sex pili are differing from a normal pili? Means sex pili are comparatively less in number than that of normal pili, and they have hollow core. The central part will be hollow, and this hollow structure is helping in the transfer of genetic material from one bacteria to other bacteria during the process of conjugation. Actually, the normal pili are useful for attachment of bacteria to the substratum, but this sex pili is useful for attachment of one bacteria to the another bacteria during the process called as conjugation. And not only it is helping in attachment of one bacteria to other bacteria, but also through this hollow core the genetic material will be transferred from one bacteria to the other bacteria. So that is from F plus bacteria to F minus. F plus bacteria means which contains the plasmid or the genome, whereas F minus which does not have the plasmid or the genophore. So likewise the F plus and F minus are attached with the help of the sex pili and helping in the conjugation, and the sex pili is acting as a conjugation tube. During the process of conjugation. So these are the characters which can be seen in the structures external to the cell wall. Those are glycocalyx, flagella, pili and the sex pili. In the structure, the next one is the cell wall. Due to the presence of cell wall, bacteria is showing the characteristic feature of plants, but at the same time it is deviating from the plants, that because it is not having the cellulosic cell wall, but this cell wall is made up of a chemical called as peptidoglycan or murines, or otherwise we can call it as muramic acid. It is also a type of polysaccharide material, and this cell wall is the structure which is made up of the peptidoglycans, and these peptidoglycans are made up of alternating structures, amino sugars. Those amino sugars are, one is called as NAM and NAG, that is N-acetyl muramic acid, and other one is N-acetyl glutamic acid. So these amino sugars are present in a alternate manner, and they have been binded with glycosidic bonds. Glycosidic bonds. So let's see the organization of this NAM and NAG organization with glycosidic bonds in a schematic diagram. NAM, NAG and NAM, like this they are in the form of a alternate manner, and this NAM has been binded to the next chain up to 4 forms. Here once again the NAM of the next chain will be present. So this got binded to the NAG, which in alternate manner, and this is binded to the alternate NAM. Here this one will be binded with the help of the glycosidic bonds. With the next group of NAM side chain, once again it is binded to the NAG and this is binded to the NAM. So this NAM is also been binded to the next group like this with the glycosidic bonds. So likewise the horizontal binding and the vertical bonding is present in the organization of the cell wall, where the NAM and NAG are in the form of a alternate side chains or alternate forms with the help of the glycosidic bonds. There is a scientist called as Christian Graham. He had developed a staining process called as a Graham staining process. So based on this Graham staining technique, the bacteria are differentiated into Gram positive bacteria and Gram negative bacteria. So what is the procedure in the Gram staining process means? Here all the bacteria have to be kept in a staining material called as a crystal violet. When all the bacteria are kept in the crystal violet, all the bacteria will acquire purple color or violet color. Then this had to be kept in iodine solution. Later it had to be kept in the alcohol, that is ethanol. So when they observe that some of the bacteria, after keeping in the alcohol, some of them are losing the violet color and some of them are retaining violet color. The one which are retaining the violet color are called as Gram positive bacteria. The one which are losing the violet color are called as a Gram negative bacteria. Likewise, the Gram staining process had able to identify or differentiate the Gram positive from the Gram negative bacteria. What is the reason why the Gram positive bacteria are able to retain the violet color, and gram negative bacteria are losing that violet color. Means here in the gram positive bacteria, they have both horizontal and vertical linking with the help of the peptide or the glycosidic bonds, whereas in the gram negative bacteria, either horizontal or vertical, any one type of linkage is present. As it have both the linkages, it forms a network like structure, and the violet color will be retained in that network like structure, whereas as they have only either horizontal or vertical linkage, the violet color will be lost from that mesh like structure. That's the reason why this gram positive bacteria are retaining the violet color. And other reason is also there, that is gram negative bacteria contains more lipids in them. These lipids, they make this alcohol to dissolve. So when they make the alcohol to dissolve, so the violet color will be lost. So that is the way how this, that is the reason why this gram positive bacteria are able to retain the violet color when compared to the gram negative bacteria. And apart from this one, the gram positive bacteria contains an other chemical that is called as a teichoic acid, so which is not present in the gram negative bacteria.